Hi, biology superstars, and welcome to the HL content for B1.2 about proteins. We're really going to get into the nitty-gritty about the form and function of proteins. So let's get started. All right, the first HL point of information deals with chemical diversity in the R groups of amino acids as a basis for the immense diversity in protein form and function. So we're really sticking with this form and function language. I think I'm going to go purple for my underlining. We talked about R groups already in the it's like a little more blue than purple, but it's fine. We talked about R groups already in the SL content. Remember, the R group is the variable side chain part of the amino acid. So you have the alpha carbon, the carboxyl group, the amine group, the lone hydrogen, and then the R group is over here, and it's different on each of the 20 amino acids. So in the HL content, we explore just a little bit more what chemical diversity looks like in those R groups. Students are not required to give specific examples of R groups. However, students should understand that R groups determine the properties of assembled polypeptides. So as amino acids fold up into proteins, the way that these R groups are is what determines what the protein looks like. Students should appreciate that R groups are hydrophobic or hydrophilic, and that hydrophilic R groups are polar or charged, acidic or basic. All right, so we saw this picture already in the SL content as well, but I really like the way that this diagram shows you the side chains of all of these different amino acids, which again, like you don't need to know, but you might notice in the non-polar ones, a lot of the side chains, okay, so like this is the amine group, this is the carboxyl group, the side chain is the thing that's up here in all of them. In the non-polar ones, these side chains are really full of carbon and hydrogen molecules, and it keeps those side chains non-polar so that the amino acid as a whole is not polar. In the polar ones, and the acidic ones and the basic ones, you'll notice that there's like other molecules at the ends of those side chains. So they're just representing them with colors here. But you'll see like this is all colored. In the acidic ones, the color specifically is all this red color, which is representing oxygen molecules. And the basic ones is this blue color, which is representing nitrogen. And that has to do with whether or not that side chain is going to want to accept hydrogen, ions or protons, and whether it's considered acidic or basic. All right, so chemical diversity in R groups has a lot to do with, like, these properties are basically what are deciding how these amino acids bond together. Properties decide bonding. That's the big takeaway from this. And then starting with the next point of information, we're going to look a little bit at how the amino acids that are in a polypeptide, how the properties of individual amino acids decide the bonding and what all that bonding is called. I think I'm going to go red for this one. All right, so the first thing we're going to look at is primary structure of a protein. So the impact of primary structure on the conformation of proteins. And students should understand that the sequence of amino acids and the precise position of each one within a structure determines the three-dimensional shape of proteins. Proteins, therefore, have precise, predictable, and repeatable structures despite their complexity. So remember, we said that the precise position of each amino acid, there's 20 possible amino acids. Hold on, I need to get my pen going. There's 20 possible amino acids, and they can be in any order at all. So we use that mathematical relationship 20 to the n to say that, like, amino acids can be anywhere along the chain. They bond together by peptide bonds. We'll put that here, maybe, bonded by peptide bonds. And the primary structure is just the order or placement of each amino acid. So for a little mnemonic device, we're going to say primary equals placement. And that means the placement of each amino acid. I'm just going to write AA for amino acid. So literally like the order that they are in this chain. Order of the AAs in chain. And we're going to, in the HL content, run through not only primary structure, but also secondary, tertiary, and quaternary structure. So the next one that we are going to look at after you think about the placement of each amino acid is the secondary structure, which is, am I going to stay with red? No, I'm going to go orange, yellowy orange. All right, so pleating and coiling of secondary structure of proteins. Oh no, I hate this color. Hold on. Okay, so hydrogen bonding in regular positions to stabilize alpha helices and beta pleated sheets. And in terms of staying with our mnemonic devices, we're going to say that secondary is stabilizing. So the secondary structure is when you have like whatever the sequence is of amino acids folding up into either alpha helices or beta pleated sheets and helices. And this happens due to hydrogen bonding between carboxyl groups and amine groups. So I'll just write it in here, due to hydrogen bonding between carboxyl and amine groups. Between carboxyl and amine groups. So remember that some carboxyl and amine groups are already bonded together with peptide bonds. There is a separate bonding force of hydrogen bonding that helps decide how amino acids start to fold up into three-dimensional proteins. A beta pleated sheet looks like this. An alpha helix or an alpha helix or alpha helices look more like that. And you can have in the same protein parts of it that are folded up into a beta pleated sheet and parts of it that are folded up into an alpha helice or helix. Oh, sorry, I'm like messing up the word a little bit. We'll go through how that happens, thinking a little bit more about the tertiary structure and when like the whole protein really starts to fold up as we start talking about tertiary structure in the next slide. All right, so here, I think I'm going to go blue pen, come over here. Blue pen, no, dark blue. Okay, so dependence of tertiary structure on hydrogen bonds, ionic bonds, disulfide covalent bonds, and hydrophobic interactions. Students are not required to name examples of amino acids that participate in these types of bonding, apart from pairs of cysteines forming disulfide bonds. And you can see those bonds here in this picture. That's why I chose this picture. This is a pair of cysteine amino acids bonded together with two sulfide molecules. That's why they call it a disulfide bond. It's happening again over here. This is a named example. You need to know this. Students should understand that amine and carboxyl groups in our groups specifically can become positively or negatively charged by binding or dissociation of hydrogen ions, and they can then participate in ionic bonding. So I think first of all, little mnemonic device, tertiary. We're going with the R to remember that tertiary structure has to do with bonding between R groups. So this is why the individual chemical makeup of each of these R groups is super important, because it's used to decide the three-dimensional shape of the protein. What else about this? The ionic bonds thing, since they name it, like maybe just as a little refresher, so the R groups can be either, let's go here, R groups, R groups can be acidic or basic. R groups, acidic or basic. And that means that they can fully give away or take on other molecules and participate in ionic bonding. All right, and the next point of information also talks about tertiary structure, so I'm going to save polar and nonpolar amino acid talk for B1.2.10. And I'll stay with blue because we're still tertiary. All right, so effect of polar and nonpolar amino acids on tertiary structure of proteins. In proteins that are soluble in water, hydrophobic amino acids are clustered in the core of globular proteins. Integral proteins have regions with hydrophobic amino acids, helping them embed in membranes. So if you have, I'm going to do like this is a lipid bilayer, right? This is the outside of a cell, lipid bilayer. If you have a protein embedded in that lipid bilayer, you're going to have a hydrophilic part. Let's make the hydrophilic part green, I think. So hydrophilic parts of the protein, hydrophilic, are going to be attracted to the water in the external and internal aqueous environment of the protein. Hydrophobic parts are going to stay here in the core of the protein, hiding inside of the lipid bilayer. And these chemical attractions are going to help the protein embed in the membrane. Like this part's going to stay inside while the green part stays outside. And that is going to mean that the protein as a whole, let's go purple for the protein as a whole, stays embedded inside of the lipid bilayer. Embedded. So that's the idea of the protein being parts, some amino acids being nonpolar and some amino acids being more polar gets important there. All right. So primary, secondary, and tertiary structure deal with just the folding up of individual amino acids into proteins. Quaternary structure, we start looking at different chains of amino acids coming together. And in quaternary structure, I'm going to go pink for quaternary structure. You can talk about non-conjugated or simple proteins and conjugated proteins. There are some named examples here. So the IB asks you to include insulin and collagen as examples of non-conjugated proteins, and hemoglobin as an example of a conjugated protein. There's a Nature of Science statement that says technology allows imaging of structures that would be impossible to observe with unaided senses. For example, cryogenic electron microscopy has allowed imaging of single protein molecules and their interactions with other molecules. We'll talk more about electron microscopy at the cellular level of organization, and sort of some of the history of microscopes. What we're going to talk about now is this idea of conjugated and non-conjugated proteins. So my mnemonic device, I'm going to delete and start over. My mnemonic device for quaternary is conjugated, which is maybe sort of a cop-out, but I think they kind of rhyme, even though I know conjugated doesn't start with a Q. And this is what the IB wants you to know. So it's helpful to just have it in the remembering device, non-conjugated. And I like calling non-conjugated proteins simple proteins, because I just think it's like easier to remember. But if you're into the non-conjugated thing, you can think non is no non-protein parts. Non-protein parts. So both insulin and collagen are composed of only amino acids. Hemoglobin also has heme groups. So there are other things besides amino acids in a conjugated protein contain non-protein molecules. Non-protein molecules. And hemoglobin is named for heme groups. You can see in this picture of quaternary structure two different chains joined together. So the chains are represented by the two different colors. So quaternary structure is when multiple chains of amino acids and sometimes other things as well fold up to form a protein. All right. And I think that brings us to our very last point of information, which is the shape of these proteins. So the relationship of form and function. I'm going to stay pink because it's the last one. Relationship of form and function in globular and fibrous proteins. So students should know the difference in shape between globular and fibrous proteins. And this is pretty easy. The globular ones are a glob, and the fibrous ones are then sort of like long and stringy, usually structural. Structural. All right. And understand that their shapes make them suitable for specific functions. Again, we're talking about insulin and collagen. So insulin is globular, collagen is fibrous. And collagen is a structural protein. So like that long strand, this is collagen here. That long stringy structure allows it to be structural. Insulin is pictured here, insulin. And insulin is a peptide hormone, and it can glob up to move around your whole body. All right, and that is where we leave the HL content about proteins. So I hope this has been helpful, and I will see you in the next video.